All right, we're going to do mitosis with pipe cleaners today. So grab some pipe cleaners or string or other objects that you have around your house, and you can follow along and help do that active learning that will make those connections in your brain with your hands and your eyes and help you understand all of this is going on. So I have chromosomes here represented by these pipe cleaners. This is going to be a diploid cell, so it's going to have two copies of each chromosome, one that came from the mother of the cell, one came from the father of each of these chromosomes. So there's 6 total chromosomes, or 2 copies of each of 3 chromosomes for diploid number. So at mitosis, these chromosomes are all going to get copied right before the cell divides. So these 2 are not the copies. These are individual chromosomes. So to make the copies, we have some more pipe cleaners that will get joined together at the centromere. This happens during the S phase of interphase before mitosis. So now our chromosomes are all copied. We still have 2 of each chromosome, and now they have their sister chromatids attached at the centromere. We still have 6 chromosomes. Now we're going to go into prophase. The chromosomes would condense and become visible out of their chromatin. Then we go into metaphase. The chromosomes are going to line up at the middle of the cell. It doesn't matter how they line up. They can line up in any way. Then at anaphase, remember that's where the sister chromatids are going to separate and go to opposite ends of the cell. So now we have 2 nuclei forming in telophase, and then the cytoplasm would split in cytokinesis, and we have 2 daughter cells that are identical copies of the initial cell that we started with. They each have 6 chromosomes that were just copied and separated in mitosis. So these are now 2 cells, one cell and the other cell. Genetically identical, nothing has changed. This is mitosis. All right, now we're gonna do meiosis with pipe cleaners. Now remember, this is the cell division that is only for sexual reproduction. This is the cell division that's going to produce the sperm and the egg cell. We're going to start with a diploid cell that has 2 copies of each chromosome, but we're going to end up with a haploid cell, actually 4 haploid cells that only are going to have 1 copy of each chromosome. So pay attention. Now, I'm also going to demonstrate for you how genetic diversity occurs through crossing over. So on one of these chromosomes, remember, since one came from the mother, we'll call that the purple beads that came from the mother. One came from the father, that will be the silver beads in this case. And we'll demonstrate how crossing over occurs to combine those traits in new ways. So we start with our diploid cell, 2 copies of each chromosome. These are going to be duplicated in the S phase of interphase. So they're going to get sister chromatids on each one. So now we have duplicated chromosomes, still 6 total chromosomes, but they have their sister chromatids attached at the centromere. Now in prophase 1 of meiosis, this is where the tetrads are going to pair up. These are the homologous chromosomes. Remember, one of these came from the mother, one of these came from the father. They have the same traits, the same characteristics in the genes that are on this chromosome, but they can have different versions of those genes. So these are the mother's versions of those genes. These are the father's versions of those genes. The tetrads of those homologous chromosomes pair up and get together, and then the magic happens. This is where crossing over occurs. So literally a piece of the chromosomes breaks off and reattaches on the other chromosomes, so we can have new genetic combinations. So now we've got a mixture of the mother and father's, now grandparents, original mother and father's traits on these different chromosomes, on these homologous chromosomes. So I'm not going to do it on all of them, but crossing over would take place pretty much on all of the chromosomes. So you get new genetic combinations for genetic diversity. Now at metaphase 1, the tetrads are going to line up at the middle of the cell. So they're still in their tetrads, but it's not going to matter which side is which. They could be lined up like this, or these could switch, or these could be switched, or these could be switched. It doesn't matter which side the maternal and paternal chromosomes are on. It's independent assortment. So that part is random, and that also gives us more genetic diversity. Now at anaphase 1, the tetrads are going to separate. So the homologous chromosomes separate. The sister chromatids do not separate yet at anaphase 1 of meiosis. Just the tetrads separate. And then telophase 1 would occur, and cytokinesis would occur, and we would have 2 new cells, 2 daughter cells, but they still have their sister chromatids. They're still duplicated. So now they're going to go into meiosis 2 to separate the sister chromatids. It's going to happen just like mitosis. So prophase 1 would occur, nothing special there. Then they line up at metaphase 2. Then at anaphase 2, the separation of the sister chromatids happens. So now the centromeres come apart. The sister chromatids go the opposite sides. And then telophase 2 and cytokinesis, and we end up with 4. Didn't go through all of these. So that would be going on in both cells, both of the original 2 daughter cells. So we would have 4 daughter cells now that have 3 chromosomes each. So they are haploid. We started with 6. Rewind and look if you need to. We started with 6. Now we have 3 chromosomes in each of these 4 daughter cells. These are going to be the sperm and the eggs, the gametes. So that at fertilization, whichever sperm and egg get together, then we end up with 2 copies of each chromosome again for the diploid embryo. All right, practice that as many times as you need to make sure you understand the differences between mitosis and meiosis.